Dr. Durairaj Sekar. I am from India, Saveetha College and Hospital, Saveetha University. So today I'm going to give a talk on microRNAs and its target in the treatment of oral squamous cell carcinoma. We are working in this field for the past 12 years, especially on non-coding RNAs. So these are all outline of my presentation. So I'm going to give introduction of OSCC, that is oral squamous cell carcinoma, and non-coding RNAs. In that, we will see microRNAs, how it originates, and what is its function, how it regulates other genes. Then whether it is a tumor suppressive microRNA or oncogenic microRNA, how this microRNA can be used as a prognostic, diagnostic, and therapeutic targets for the treatment of oral squamous cell carcinoma. Then, of course, future perspectives and conclusion. So let me talk about OSCC. That means, in nutshell, oral cancer is the 16th most prevalent cancer, and it's a silent killer. So we don't know how it happens sometimes, with approximately 300,000 new oral incidents reported each year worldwide. Oral cancer is a form of head and neck cancer. We know that, with the most common subtypes being oral squamous cell carcinoma. So this is the subtypes of oral cancer. The major risk factor for OSCC is chewing, smoking, alcohol consumption, maintaining poor oral hygiene, and most importantly, infection of human papilloma virus. So it happens because if we have human papilloma virus, then of course there is a high risk of OSCC. So what happens is, although much progress happens nowadays to treat these deadliest diseases recent years, but we don't have a therapeutic targets, proper therapeutic targets, and most of the drugs that are currently useful to treat this disease has side effects, and it's very challenging to manage this disease with these drugs. Unfortunately, the patient survival rate has decreased to less than 5% due to poor prognosis, and we don't have poor prognosis. So even in developed world, there is no proper diagnostic and prognostic method. So therefore, scientists are thinking that new approaches are urgently needed. That means new molecules are needed to target this disease. And everywhere, even developing countries, there are so much of population, and we cannot control this disease even with the newly identified drugs. So we need some kind of new molecules, exact molecules that can revert the cancerous cell as a normal cell. So coming to the non-coding RNAs part. So non-coding RNA is the biggest part, or the functional RNA molecules that are not translated into the protein. That is called non-coding RNA. So basically, so the number of non-coding RNA within human genome is unknown, because we don't know how much is the non-coding RNA which is present in the cells. Recent transcriptomics and bioinformatics studies suggest that there are thousands of non-coding transcripts. That means we don't know what happened inside the cells. We know genes. There are plenty of genes and plenty of proteins. There are so many signaling pathways. But apart from that, this non-coding RNAs plays major roles to control signaling molecules and our signaling pathways. So these regulatory non-coding RNAs are two types. One is long non-coding RNAs. The size is 200 nucleotide in length or more than that. The second one is called small non-coding RNA. The size is about 16 to 22 nucleotide in length. There are three types of small non-coding RNAs. One is called microRNAs. The second one is called small interfering RNAs, that is called siRNA. We know that. The third one is piRNAs. So we are going to concentrate on microRNAs because that is our part. We are working on that, and we are curious to understand how these microRNA regulating various pathways to suppress various kind of signaling pathways to induce the disease progression. So what is microRNA? So microRNA, most of the studies, we know that what is this microRNA? So small RNAs, what it can do in the cells. So it is highly conserved, it's very small. So as I said, it's 16 to 22 nucleotide in length. It's an uncoding RNA, it's not code for any protein. But these molecules involved in the regulation of gene expression by binding at the 3' untranslated region. Sometimes, there are so many controversies. They may bind to 5' end or 3' end, but most of the microRNA, they can bind to 3' end because there are some complementary sequence or some seed sequence that is present in the target gene. In other words, each and every microRNA has its own target. That means in the 3' end, it has a seed sequences or a complementary sequence that can easily, this microRNA can easily go and bind and stop the translation or modify the it acts as a post-transcriptional modifier. So the second interesting thing is this microRNA present in the body fluids like plasma, saliva, urine, and other kind of body fluids. But what is the use? Since it's present in all kind of body fluids, it can be used as a prognostic molecule or diagnostic molecule. So easily detectable in the sense. So in the cells, there are two kinds of pathway in which this microRNA can be originated. One is called canonical pathway and another one is called non-canonical pathway. So as like a gene, this microRNA originated from the nucleus and entered into the cytoplasm. In cytoplasm, it is matured and then it binds to the targeted gene. This is what the function of microRNAs in the cells. So after binding, what happened? It may degrade the targeted RNA, that's called mRNA, and it can inhibit the translation and regulate the various gene function. This is the function of the microRNA in general. So for example, let me explain the canonical pathway. It's very simple. So in the nucleus, as like a gene, as I said, this microRNA, it is originated from the gene through RNA polymerase 2, which is called primary microRNA. This primary microRNA is processed by DROSHA, the protein called DROSHA inside the nucleus is processed. And then the processed microRNA is called precursor marker or pre-microRNA. These precursor microRNA are exported to the cytoplasm through exportin 5. This is the transporter protein, which is present in the nucleus, inside the nucleus, and it is the pre-microRNA can easily transported via exportin 5 protein. Then this precursor microRNA again processed in the cytoplasm by dicer is another kind of protein which is present in the cytoplasm, and there are so many researches going on the interaction between dicer and the precursor microRNA. If you stop this dicer molecule, we can able to suppress some of the microRNA so that we can save the cells. This is another concept is going on. Then after dicer processing, microRNA duplex, that is the secondary structure, can be processed, and then the microRNA can be a single strand, which is called matured microRNA. The microRNA duplex, which are loaded with RISC complex, RISC is nothing but RNA-induced silencing complex, can go and bind to the target gene. This is called canonical pathway. So if you see this pathway, if you block any of this pathway, we can easily able to able to stop the microRNA biogenesis. In this way, we can save the cell if the microRNA is oncogenic. So what are all the activity of microRNA? One is tumor suppressor. I mean, it acts as a tumor suppressor. The second one is which may also act as an oncogene. So microRNA have been identified to act either tumor suppressor or oncogene. We already know that. So what scientists have discovered that tumor suppressor microRNA whose expression is down-regulated in tumors. In other words, oncomeres are, that is oncogenic microRNAs are upregulated. So these are all some of the microRNAs that are used, that are not currently used, but it's still validated through preclinical studies. So microRNAs and its target as a prognostic and diagnostic biomarker for OSCC. For example, microRNA 21, we know that this 21 is involved in all kinds of diseases, including OSCC, which is easily detected in plasma and also saliva. So it is always upregulated in the cells and target MAPK1. So MAPK pathways can be targeted by microRNA 21. The other microRNA is 548B, which is easily present in, which is easily detected in tissues, which are upregulated. And then the targeted gene is ACTN4. Likewise, each and every microRNA has its own targeted gene. So if it is upregulated, that microRNA we may call oncogene, because what happens is if it is upregulated, it will go and bind to the tumor suppressor gene. So that what will happen is the the cells become tumor cells. The second type of microRNA is called tumor suppressive. So this is always down-regulated in the cells, and it has own targeted gene, which is also easily detected in saliva or blood. For example, microRNA 24-3P and microRNA 155. These are all microRNA are detected in the OSCC samples, according to the recent studies. So other microRNA, which is called therapeutic molecules. For example, previous one we have seen some molecules are detected. We can use as a diagnostic or prognostic molecule. So if you want to test whether you have disease or not, you can go and detect this microRNA. If the upregulated microRNA is present, the disease is present, or otherwise it's not, like that. The second kind of microRNAs is called therapeutic molecules. There are so many microRNA are detected, which are identified recently in the tissue as well as in the cell lines. And most of these microRNA are not validated in the clinical studies. This is very important because the first line study is cell lines. The second line is preclinical validation. And the third one is clinical validation. Until now scientists are struggling whether these microRNAs are really involved in the cellular pathways and really we can use as a therapeutic or diagnostic molecules. For example, microRNA 221, so which is present in the OSCC tissue, which are always upregulated, and the target gene is PI3K hormone. So these are all some of the microRNA that is involved in the OSCC that we see. This study, recently we have published this from our lab. The microRNA, which is called microRNA 138, it's a therapeutic molecule for oral squamous cell carcinoma. So what happened is when we used to screen the microRNA, lot of microRNA, we have seen some of the microRNAs are upregulated, some of the microRNAs are downregulated. So we have picked up the most microRNA that is present in the cells, and then we have seen the pathways which is related to this microRNA. We are still studying this. We are curious to study this, and then until now we are not yet concluded correctly. We are still in progress. Our experiment is going on. These are all some of the publications we have done until now. MicroRNA 138 and MicroRNA 185 is also detected. The most important thing is each and every microRNA has its own, we can synthesize antimer, that is antagomer, we can call antagomer. That is each and every microRNA, there is a sequence. We can synthesize the opposite sequence so that we can block this microRNA in the cells, which is called anti-microRNA. We have tested so many anti-microRNA against few microRNAs and see how it works, whether the cells is saved, how the experiment, we are testing all kind of experiment. Our studies is going on. Coming to the future perspective. So as I said, we need a lot of validated preclinical studies, and it's still going on. So microRNA based therapeutics like anti-microRNA or microRNA mimics. So we need it. And also, sorry. Sorry, I'm sorry, little bit disturbance. So microRNA based therapeutics like anti-microRNA or microRNA mimics. This anti-microRNA is opposite to microRNA mimics. So some of the down-regulated microRNA, you can pick that and you can synthesize and you can insert into the cells and see the effect. For example, microRNA can be loaded in the exosomes or nanoparticle or liposomes and can be used for the successful delivery to a target site. The future studies characterization of disease-specific markers or on targeted tissue and explore new targeting ligands for improving microRNA therapeutic efficacies are needed. So in conclusion, as I said, more validated studies are required to establish microRNA as a potential biomarker and therapeutic targeting OSCC. So thank you very much. If you have